Hi guys, my name is Jamil and today we are going to start about biochemistry. And biochemistry first chapter is about nucleic acid structure and organization. The first topic of nucleic acid structure and organization is the central dogma. So you know that every organism has genetic information. He stores these genetic informations and also might transfer these genetic informations for the preservation for the preservation, and he also transferred these genetic information for the future generation. So for the future generation, and he did all of these things, genetic information is stored and transferred to future generation with the help of one of the major steps known as the central dogma. Now first of all, know that what is the central dogma. Central dogma consists of two things. The first one is that replication and gene expression. Central dogma consists of two things. First is the replication, and second is the gene expression. A gene expression further consists of two things. First one is the transcription and translation. So it was about central dogma. The central dogma consists of two processes: replication and gene expression. And gene expression further consists of two: transcription and translation. So this whole is the central dogma. Let me explain what is the central dogma in detail. Let's suppose that in central dogma, when there's a DNA, in the DNA there may be the replication, means that I'm going to tell you that what is the replication. Replication is the DNA will produce its own copies, mean that its blueprints will produce, chromosomes will be produced, duplicated chromosomes will be produced in the replication. So what is the replication? Replication is the production of duplicated proteins. Duplicated chromosomes mean that duplicated blueprints of DNA will produce in replication. So it was about replication. And second is the gene expression. You know that gene expression is from here to here. These two processes involve gene expression. One is the transcription and second is the translation. First was the replication, it's okay, and second is the gene expression. Gene expression consists of two things. The first one is the transcription, second is the translation. So what is the transcription? Transcription is the production of RNA blueprint from the, it will produce the RNA copies from using the DNA. So it will, during this process, DNA will be used and it will produce the RNA. So what is the transcription? Production of RNA from using the DNA. And second is translation. Translation is the formation or production of the proteins from RNA, mostly messenger RNA. I mean the messenger from messenger RNA, the production of proteins is the translation. So it was about central dogma. Central dogma consists of replication and gene expression. What is replication? I have told you. And what is the transcription? What is gene expression? It's the two things: transcription, production of RNA from DNA, and translation, production of proteins from DNA. So it was about central dogma. But here is some opposite process to central dogma. I'm going to tell you about that this is the reverse transcription. This is an exception case. This is the opposite to the central dogma, or we can say that this is opposite of transcription. Reverse opposite transcription means that it is opposite the transcription. In this case, a DNA will be produced from RNA. Using the RNA, DNA will be produced, and this mechanism mostly happens in retroviruses. Mostly happen in retroviruses. Retroviruses has ability to produce DNA by using the RNA with the help of enzymes named as the reverse transcriptase. Reverse transcriptase. These retroviruses have enzymes named as the reverse transcriptase. This reverse transcriptase will catalyze the reaction, the production of DNA from RNA. It will catalyze the reaction named as the reverse transcription. So retroviruses has ability to produce the DNA from RNA. This reverse transcriptase enzymes also called as the RNA directed DNA polymerase. RNA directed DNA polymerase. So we can say that reverse transcription is the opposite of the central dogma. In this case, DNA will be produced from RNA, and this process happens in retroviruses with the help of enzymes or reverse transcriptase. Reverse transcriptase also it's also named as the RNA directed DNA polymerase. So it was about the central dogma. Basically, what is central dogma? Now we are coming here. We have used two terms. First is the replication and second is the gene expression. So we still here we saw about their definitions, but what is the basic difference between these two terms? That what is the replication and what is the gene expression? What is the basic difference between these two things, these two terms? The first one is types of nucleic acid difference. Mean that in replication there may be different nucleic acids, and in gene expression there will be different nucleic acids. Mean that nucleic acid may be different. Replication has different nucleic acids, and in case of gene expression there will be different nucleic acid. Mean that the ribonucleic acid and deoxyribonucleic acid, there will be change. In RNA, ribonucleic acid, in DNA, deoxyribonucleic acid. The second part is length of nucleic acid. Length of nucleic acid will be different. You know that a single chromosome have, let's suppose, this size. So when it will be replicated during replication process. When it will be replicated, then it will produce the same copy, duplicate, and it has the same length, like that. Same length. But in case of RNA production from DNA, mean that during gene expression, it was about replication. But in case of gene expression, when gene expression happened, that RNA will produce from DNA, but RNA will produce small little, like that, like a little drop. So it has a very small size. So this is the DNA. You know that DNA has a large length and RNA has smaller length. So this is also difference that in replication length will be greater. It is going to be 10 raised to power 8 nucleotides pairs. But in RNA these are 10 raised to power 4 to 10 raised to power 5 nucleotides pairs. So what is the difference? The difference is that in the replication DNA has a greater length, but RNA has a small length as compared to DNA. These are the size of 10 raised to power 4 to 10 raised to power 5 nucleotides pairs, but DNA has 10 raised to power 8 nucleotides pairs. Mean that DNA has 2 to 3 times more length as compared to RNA. So it was a second difference between replication and gene expression on the basis of length of nucleic acid. The third one is about enzymes involved difference. There are different enzymes involved. You know that in the replication, which enzymes involved? The enzyme involved is the DNA replication. That enzyme name as the in DNA replication, enzymes involved as the DNA polymerase. DNA polymerase. And in gene expression, enzyme involved is the RNA polymerase. RNA polymerase. Why? Because in replication we have to produce the deoxyribonucleic acid. So the enzyme involved with the DNA polymerase. But in gene expression we have to produce the ribonucleic acid. Look at this. We have to produce the ribonucleic acid. So therefore the enzyme involved with the RNA polymerase. In replication we have to produce the deoxyribonucleic acid copies. So therefore enzyme involved with the DNA polymerase. But in transcription, gene expression, we have to produce the ribonucleic acid, ribonucleic acid. So therefore enzyme involved with the ribonucleic acid polymerase. And the further is the proteins formation, and this is also a difference between these two things. During gene expression we will produce the proteins. And what is the function? What is the end product of gene expression is the protein formation, and what is the end product of the replication is the chromosomes duplicated. Means that in this process chromosomes will be duplicated, but in translation, in gene expression, the product is the protein. So it is involved in protein formation. What protein formation involved in gene expression, but chromosome duplication involved in a replication process. And one more important thing about this, that because every organ, our every tissue have a different kind of proteins. Let's suppose that liver has a different proteins, stomach has a different protein. I mean that every organ has a different proteins. So it will produce, gene expression will produce all kind of different proteins for different organ. But in case of chromosomes, you know that chromosomes are same in our body. So during application, same chromosomes will be produced, no different chromosome, because all our body, every cell has the same chromosome. So during application, same chromosomes will be produced. But in case of protein formation, you know that every organ has different proteins. So gene expression will produce different proteins for different organs. So it will look different between protein formation and chromosome duplication. And second is that this will cytoplasm. Gene expression will always happen in a cytoplasm. You know that translation will always happen with the help of ribosomes, and the messenger RNA will produce protein. It will happen in cytoplasm. So translation will happen in cytoplasm. But in case of actually replication, you know that replication happens in a nucleus. But we can say that transcription also happens in nucleus. Transcription also happens in nucleus. Replication happens in nucleus, and the second part is that translation happens in cytoplasm. It will never difference between that. I'm going to tell you another point about the length of nucleic acid. I forgot at that time, but now I'm going to tell you that length of nucleic acid. You know that when we take the exam, when we examine the DNA, then we have to cut the DNA into different pieces, into smaller pieces like this. We cut the first DNA into smaller pieces for examination, but RNA will not be cut down into smaller pieces because this is already a small piece, already a small 10 to the power 4 to 5 nucleotides pairs. But DNA has a larger length, so we have to cut down the DNA into smaller pieces with the help of special enzyme for the examination. But RNA is already a little length, so we can use the RNA as a whole because they're already a little particle. The last difference between these replication and gene expression is also most important. That phase of cell cycle difference. Phase of cell cycle difference mean that gene expression will happen throughout the interphase. It will happen throughout interphase, and this replication will happen in S phase. The point is here that gene expression happens throughout interphase. During whole interphase, gene expression will happen, but replications happens just during S phase. To cover this point, first we have to cover what is the cell division. So come here please. What is the cell division? To cover this point, this last one point. We're going to cover this last one point. So please come here. Go to cell division. You know that cell division consists of two things: mitotic phase and interphase. Mitotic phase is the production of two daughter cells from one to two daughter cells. Mitotic phase. And in interphase consists of a different phase. It is a time between two mitosis. We can say that between two mitosis, between two cell divisions. So interphase is the time between two mitosis, and mitosis is the 1 to 2 daughter cell production. So let me explain here in the figures, in the form figure. Suppose that this is the whole cell cycle, and you know that cell cycle consists of two things: a smaller one, for a small period, mitotic phase, and larger period still here to here, and it is still here to here is the interphase. Yeah, look at this. This is the interphase. So smaller mitotic phase and greater is the interphase. Let me explain first what is the cell division. You know that. First, we will start from the G0 phase. You know that some cells go G0 phase. Also, we can say that resting stage or non-dividing stage. Resting, we can say that resting stage are non-dividing. So G0 is the resting state or non-dividing state in which some cells go and they do not undergo a cell cycle. And these cells, you know that nerve cells and muscle cells, examples of the G0 phase, they undergo G0 phase and they will not divide at all. They will undergo resting cells. Example, nerve cell and muscle cell. Remember, very important. Very important is the nerve cell. Nerve cell is most important. So nerve cell or the resting cell, a non-dividing cell, they will undergo the G0 phase. But some cells go back from G0 phase to G1 phase, which is the phase of interphase, which is the first part of the interphase. G1 phase, S phase, and G2 phase. All three are the further phases of interphase. So interphase consists of how many phases? 3. G1 phase, S phase, and G2 phase. So some cells go from G0 phase to G1 phase. Examples are the epithelial cells. Epithelial cells will undergo G1 phase and they will start their division. So let's suppose that now they have come in the G1 phase epithelial cell. In G1 phase, cell will prepare itself for the cell division. In G1 phase, what will happen? Cell prepares itself for the replication. For the cell division. So now, from G1 state will active the S state. But how this S phase will be active? With the help of signals. with the help of signals, and these signals comes from growth factors. These signals comes from growth factor. Which growth factor? Let's go that insulin growth factor, heparin growth factors. I mean, like when G1 phase will be G1 phase is the preparatory phase. Cell will prepare itself for the cell division. After G1 phase, S phase will be activated. But how? With the help of signals, growth factor signals, and these growth factors may be the insulin growth factors, heparin growth factors. Now S phase has been activated. Remember that. No, I'm going to tell you that replication happens in S phase. So S phase is specially pointed for the replication of chromosomes. Meaning that here chromosomes will be duplicated. So S phase is the phase in which replication will occur. And second is the G2 phase. G2 phase is the phase before the mitosis. Meaning that it's a state after that cell undergoes cell division. So it is a whole interphase, G1 phase, S phase, and G2 phase. And remember that I have already told you that gene expression happens throughout interphase. So from here to here, the whole will happen the gene expression in 3 phases. But replications occur just in S phase. So replication will occur in S phase. Next start the mitotic phase. After interphase, start the mitotic phase of a small period. In this case, one cell will form 2 cells. So it was the point that gene expression occurs throughout the interphase, and S phase, replication will occur just in the S phase. So now I would like to give some clinical correlation of about the central dogma, and by using these phases, and we can treat some diseases like cancer. Let's suppose that we use chemotherapeutic agents target specific phases. Chemotherapeutic agents mean that drugs. We will use the chemotherapeutic drugs by targeting specific phases. Mean that for each phase we have a specific drug. We will attack on the specific phase by the specific drug. Then we will block that phase. When that phase will be blocked, then the cell division will be blocked. When cell division will be blocked, so that cancer will be cured. We can cure the cancer because cancer is uncontrolled division of the cells. Then cell division will be controlled, then cancer will be controlled. And how can we control? We control by specific drugs which acts on the specific phases. Then specific phase will be blocked, then cell division will be blocked, and cell division will be blocked. That uncontrolled division of cell division will be controlled, and we can control the cancer by this method. Let's continue. That chemotherapeutic agents target specific phases. For the S phase, we have mostly 2 important drugs. The first one is the 5-fluorouracil. 5-fluorouracil. It's important. And second is the mitosis. It's also important drug. So what's the one that that's about to remember that for the S phase specific blocking, we use the 2 specific drugs, very important drugs, 5-fluorouracil and methotrexate. But we must know that what is the mechanism of these drugs to act. Let me explain their mechanism here. First, we'll discuss about the 5-fluorouracil. You know that simply in the cell division there is an enzyme named as the thymidylate synthase. What is the function of this thymidylate synthase? Thymidylate synthase will change the deoxyuridine monophosphate into deoxythymidine monophosphate. What is the function of that? It's simple. In cell division, we have in S phase. During S phase, we have a simple enzyme named as the thymidylate synthase. This enzyme will cause the conversion of deoxyuridine monophosphate into deoxythymidine monophosphate. But when there's a cancer, there is an uncontrolled division of a cell, then we'll give this drug 5-fluorouracil. When 5-fluorouracil will give, it will block the thymidine synthase. When thymidine synthase will be blocked, then the conversion of DUMP into DDMP will not happen. When this conversion will not happen, means that DUMP will be increased. Deoxyuridine monophosphate will increase. When the deoxyuridine monophosphate will increase, then it means that cell division will be damaged. Cell division will be damaged, so cell will not divide, and cell will not divide. Then by this way, we can control the cancer. And this drug is especially used for cancer. So, example, we can use it for the cancer. So it was about the mechanism of 5-fluorouracil. Also, the mechanism of method reset. Method reset has its own mechanism. Method reset, that's in common. First, we know that in common there is a dihydrofolate. There is a dihydrofolate reductase enzyme. Reductase enzymes. Also, in the case of synthesis, what is the function of dihydrofolate reductase enzyme? It will cause the chainment of the dihydrofolate into tetrahydrofolate. Meaning that it will cause the cyclic balance of folate. It will control the cycle folate. It will control the cycle of folate. It will change the dihydrofolate into tetrahydrofolate, this enzyme. But when there is a cancer uncontrolled cell division, then we give the method reset. This drug will go, and this drug will block the dihydrofolate reductase. When this enzyme will be blocked, then this cycle will be disturbed. When this cycle will be disturbed, you know that folate is important for the cell division. When folate cycle will be imbalanced, then cell division will be imbalanced. When cell division will be imbalanced, then cell division will not happen. So cell will not divide, so we can control the cancer. But this drug is not only used for the cancer, we can use this drug also for the rheumatic arthritis. Rheumatic arthritis, and we can also use this drug for the cancer. So both this drug can be used for both cases. And second is the G2 phase. For G2 phase, we use the special drug name, that is the bleomycin. That name is the bleomycin. And bleomycin is used for the cancer, is used to treat the cancer. It is used to treat the cancer and Hodgkin's lymphoma. I would like to write Hodgkin's lymphoma, and also the non-Hodgkin's lymphoma. Bleomycin is used for the cancer treatment, Hodgkin's lymphoma, and non-Hodgkin's lymphoma. We can use this drug bleomycin, but bleomycin will especially act on the G2 phase. Remember that bleomycin will especially act on the G2 phase, and we can give the bleomycin through intravenous with the help of injection, or intramuscle or intravenous with the help of injection. It will treat these diseases, but there is also an exception that bleomycin also can cause the pulmonary fibrosis. It can cause inflammation in our lungs. It can cause the inflammation in our pulmonary system, pulmonary fibrosis, because this drug may be followed by the inflammatory cells and edema. Because this drug will be followed by the inflammatory cells and edema. Remember that when inflammatory cells and edema will come, then it will cause the inflammation, and that inflammation named as the pulmonary fibrosis. This is followed by the bleomycin. That side effect of bleomycin. But it is used for the cancer, Hodgkin's lymphoma, and non-Hodgkin lymphoma, and especially acts on the G2 phase. Another phase is the M phase. We use special drugs which blocks the M phase, and that names as the prostoxyls, also called toxyl simply, plus toxyl. We also simply say the toxyl also, and there's a vincristine drug, and there is a vinblastine drug. We can use these drugs. We can use these drugs which will specially act on the meiotic phase, and it will block the meiotic phase. When meiotic phase will be blocked, then meiotic phase will be blocked, then cell division will be blocked. When cell division will be blocked, then we can cure the cancer, especially for the cancer. So, what is the mechanism of these drugs? Prostoxyls, vincristine, and vinblastine. These drugs will cause, like explain here, you know that during cell division, there's a spindle formation, like there's spindle will be formed in normal case. with the help of macrotubules. These spindles are formed with the help of macrotubules. But when we give these drugs, then these drugs will go and they will cause the dispersion, falling apart of the macrotubules. Macrotubules will not form. When macrotubules will not form, and spindle fibers will fall apart. And spindle fibers will fall apart. Then no cell division will not happen. Cell division will not happen. Then we can control the cancer. And same vincristine and vinblastine have the same mechanism, that they will prevent the assembly of the macrotubules. When macrotubules will not form, the clear down means that spindle fibers will fall apart. When spindle fibers will fall apart, then there is no cell division. When no cell division, then we can cure the cancer. The next is these were some specific drugs which we use for the specific phase, but we have also non-specific drugs which will act on the whole cell division. These are not specific drugs. These are used for the leukemia, all the kinds of cancer, especially for the cancer. So this is used for the leukemia. Non-specific drugs will use for the leukemia, but they have the same mechanism, that they will go and they will stop the cell division. When the cell division will stop, then we can treat the cancer. We can treat the leukemia, but it's non-specific. It is not specific. But these S phase, G2 phase, M phase, are some specific drugs which acts on it and give their specific function. And I would like to tell you that in S phase, 5-fluorouracil and methotrexate is important, but there is also another drug we can use, that named as the hydroxyurea. Hydroxyurea is also a good drug that we can specific target the S phase. So it was about the first topic of the first chapter named as the nucleic acid structure and organization, and first topic was the central dogma. So that is about central dogma, and in the next lecture we will discuss about the nucleic acid structure and nomenclature. So thank you so much, guys. Bye.